We're going to have a chat right now from Dr. Gabi Hanna about faster no's and accelerated yeses.
Hi, everyone. Joanne asked me to put like short presentation to be translational. Since we're talking about translational research, so you are perfect translational between our morning science sessions and the money people. So, so it will be short presentation, and I thought we will target about advancing translational research, and the heart of it is when you say no very fast and how to accelerate yes. So I will start with the second, highlight this with the second major factor why clinical trial fail is inadequate preclinical work. Major factor number one is human model. Animal model is not a human model. So, but no, we cannot do anything about it. Number two is poor preclinical work. And we can go on and on with the reasons, but I will visit back this point and to see why. There is multiple study done and led by big pharmaceutical like AstraZeneca, Amgen's about reproducibility of preclinical trial. And both studies, they found reproducibility is between 11% to 28% only. So that's if you can produce your work in animal, which is you build your all science and translational point about to take your product to clinic, you can produce it only at 11 to 20%. So how are you going to expect to have result in a human? So that is the kind of the heart of it. Now, more recognizably by NIH and all this pharma, now they trying, after they perfect the clinical trial zone, how we can do it faster, more efficient, and arguably more expensively also to save time. So now they think it is, we have to fix the underlying issue, which would comes to the preclinical, and how you're going to decide which one go and which one no go. And especially now we are in age, all pharma and a lot of universities sitting on hundreds of molecular, and they don't know which one is going to be perfect one, because all this technology, you have a slightly modifications, and you don't know which one you're going to pick. So go visit about the 11% reproducibility, and just gonna go deeper, you're going to see why this 11% is there. So in analysis of hundreds of paper that AstraZeneca did, it was shows only 2% of the paper the treatment was blinded. So you would expect, like, this is standard for clinical trial. And now in preclinical, 98% of the treatment is done unblinded, and every one of us is biased to their research, biased to, I mean, you wouldn't start company, you wouldn't do research unless you are passionate about it. So, but the issue, you can't deny your bias, but people should recognize their bias, and we should put mechanism in it. And in a clinical trial, we have to have blinded study to eliminate, some way to eliminate this bias. So in a preclinical, it's around 98% non-bias. So randomization of treatment. So this was the other bigger issue. It was 73% of the study, they don't have randomization of animal and treatment. So that is like, in clinical trial space is, blind study, blind misrandomization. This is like first things you do. So that's as one of these non-good practice. The other two, there is long list, but just I thought we'll highlight few. The number of animal, will comes to how much waste in money and time and animal we have. Like we have more than half of the studies we done in here in research, either industry and academia, half of the study, they don't have the correct number of animal. And actually third of them, they use more animal and taking more time, which time is sometimes much more valuable, and most of the time actually. And estimately, just that was number I started by, I thought we'll list it. In the US only, every year we use 500 million animal for research. 500 million animal for research. Just the cost of animal without housing, without experiment, the cost of animal estimately comes to $50 billion. Just this is the cost of animal. Just that is give you perspective how much waste and things done inefficiently. For me, for me, I'm physicians. Usually I look for things when I do any research, what is the end game? Where do you want to go? And when you see kind of things working in cycle, just wasting animal and do things and doesn't go. So that is kind of the current status. For sake of time, I wouldn't go to the solutions, how we do things, but just from this number, and everyone recognize who knows the field now, especially in recent years, it has to change. How we conduct preclinical research, it has to change, because our success rate is very poor. That's contribute to how much our drug development is very expensive. It's prevent a lot of startup with a good opportunity doesn't go forward because your risk is very high to fail, and also you will be measured with the others. So there is a lot of opportunity we can develop because of these numbers, and a lot of bad products shouldn't go to clinic, they go. So I wouldn't go in details in the solution, but in generally, I think the solution, it has to come in more comprehensive approach to preclinical and translational research. Traditionally, usually researchers are focused on what they know on their field, and they are very micro about what they know. But the drug development and translational, you need the clinical relevantly, you need the regulation, you need not only the cell culture, you need to know the animal part, the market analysis. So you need to have more comprehensive and connectivities, and you put all these things together as you move forward. And you have to have more supportive system. It's not like someone say, okay, maybe I have something good, and then they go look for solution and what is the pathway to go translational. So those kind of this solution, what I think what gonna change. And the good things this year, NIH start focusing on that, and they, for their grant applications, now they focusing on how you address this problem in preclinical when you have translational project. Big pharma now they focusing on that. Kind of they know they have to change. So the old way of how we do animal research, it has to change, and now it is time to do it. Thank you again, and glad you.
Thank you.
I'm on time.